Good morning, everybody. Welcome back to daily lectures by New Age Cosmetic White, EdTech Healthcare Consultancy. I'm Dr. Bobby, and we are on the subject of molecular biotechnology, and we are on the topic of vaccines. We continue with the topic of interest being a milestone note on the protection against foot-and-mouth disease by immunization with a chemically synthesized peptide predicted from the viral nucleotide sequence, courtesy and thanks to the authors J. Bittel, R. A. Horton, H. Alexander, T. M. Schenek, J. G. Sutcliffe, R. A. Lerner, D. J. Rowlands, and F. Brown. It's published in Nature 298-30-33, 1982. Thanks for this wonderful and invaluable note. Now, on reading the note, we see that since Jenner developed the first vaccine over 200 years ago, most human vaccines against viral diseases have included partially killed or attenuated preparations of the disease-causing or a similar non-pathogenic version of the virus. While this approach has been undeniably effective and prevents the spread of a number of viral diseases, it is clearly limited. For example, not all viruses can be grown in cultures, which precludes the development of vaccines against these viruses. Production of traditional vaccines is expensive and potentially dangerous, and not all viruses are preventable through the use of these traditional vaccines. With the advent of molecular biotechnology, alternative strategies were examined for developing safer, less expensive, and more effective vaccines that would not have the limitations of using whole viruses, killed or attenuated as vaccines. Since vaccines immunize individuals by priming the immune systems, it was thought that for some viruses, short synthetic peptides might elicit the same antibody response as the antigenic determinants normally found on the external surface of the virus. These peptides were designed with the identical linear sequence of amino acids that made up the viral antigenic determinant in the first place. Of course, this approach could be expected to work only when the amino acids of an antigenic region epitope were contiguous. Bittel et al. Isolated and characterized the viral RNA for foot-and-mouth disease virus, and that's FMDV, and then determined the sequence of VPI, the major antigenic protein of the virus. Based on other experiments, they reasoned that the major antigenic determinants of VPI, VP1, would probably be found at either the N or the C terminal end of the protein. They then chemically synthesized a series of peptides based on the amino acid sequences of the N and C termini, chemically linked these peptides to carrier proteins, and then used them to inoculate rabbits and guinea pigs with peptides from the C terminal end of VP1 as antigens. The treated animals synthesized antibodies that reached them against disease from the whole foot-and-mouth disease virus. This work established the principle that a protein domains is sufficient to induce antibodies that can neutralize intact virus particles, and hence a new type of vaccine which after the initial stages of development does not utilize or depend upon the disease-causing virus is possible. This is figure 1216, which shows the use of non-pathogenic S. flexneri to deliver foreign DNA to mammalian epithelial cells. A strain of Shigella with a deletion mutation in the ASD gene, which encodes the enzyme beta-semialdehyde dehydrogenase, is unable to proliferate and can be used as a live vector. And now the vaccination may be delivered orally, greatly simplifying the delivery of a variety of vaccines. Some limitations of plasmid-based vaccines are: 1. The necessity for strong promoters that function in vivo to selectively transcribe the induced DNA. 2. Low levels of foreign gene expression resulting from differences in codon usage between the introduced gene, often of viral, bacterial, or parasitic origin. and the animal being inoculated, and 3, the presence of antibiotic resistance gene marker genes, genetic marker genes on the plasmid vector. To avoid the use of antibiotic resistance marker genes, researchers have developed a series of minimalistic, immunogenically defined gene expression MIDGE vectors following insertion of the gene of interest into a MIDGE. This figure shows the use of a MIDGE vector to produce a capped linear DNA sequence containing the gene of interest, a promoter, an intron which facilitates expression of the gene of interest, and polyadenylation signal. Shows antibiotic resistance gene with excision side and excision side on both sides. We have promoter plus intron, gene of interest, and polyadenylation signal. An addition of oligonucleotide cap. How we proceed to the matter proper, where following the insertion of gene of interest into a MIDGE vector, the antibiotic resistance gene is excised from the vector, and an oligonucleotide cap is added specifically to both ends of the linearized DNA that contains only a promoter, intron, and a gene of interest and a polyadenylation signal. The remaining portion of the plasmid is degraded by exonucleases. The capped ends are resistant to exonuclease digestion. The purified capped linear MIDGE vector is then used directly for transvection. These vectors have been used unsuccessfully as a substitute for plasmid vectors. Vaccination of rhesus macaques monkeys with the DNA encoding simian immunodeficiency virus, followed by a booster with a modified vaccinia virus that encoded many of the same proteins, protected the monkeys against infection by simian immunodeficiency virus. The DNA that was infected at 0 to 8 weeks expressed the simian immunodeficiency virus proteins GAG, POR, WIF, VPX, and VPR, as well as the HIV type 1 proteins ENV, TAD, and REB. The recombinant vaccinia virus is expressive simian immunodeficiency virus proteins GAG and POR, and the HIV protein ENV, all under the control of vaccinia virus promoters, and was administered at 24 weeks. The protection against simian immunodeficiency virus at 7 months after the booster with the recombinant modified vaccinia virus was much greater than protection with either treatment by itself. This procedure also confers immunity against mucosal viral challenge. This feature is important because the right site of entry of the virus into the simian host is effectively blocked. It is also noteworthy that the immunity was maintained for a long time. Also, dental caries, the gram-positive facultatively anaerobic bacteria Streptococcus mutans and Streptococcus subrenus are considered to be the primary causative agents of dental caries tooth decay. These organisms colonize tooth surfaces and metabolize sucrose to produce lactic acid, which causes the enamel to become vulnerable to decay. Sucrose is also used to produce a sticky extracellular dextrin-based polysaccharide glucan that facilitates Streptococcus cells adhering to one another and to tooth surface, forming plaque. It is the combination of plaque and acid that leads to tooth decay. Two regions of one of the adhesion products found on the surface of S. mutant and S. subrenus cells are important in the adherence of these bacteria to tooth surfaces. One sequence is rich in alanine residues, while the other is rich in proline residues. Another important component of the mechanism of tooth decay is the enzyme glucosyltransferase, which is responsible for the synthesis of glucan, an insoluble extracellular polymer. This figure shows vaccination regimen of rhesus monkey with DNA containing simian immunodeficiency virus, SIV genes, and vaccinia virus carrying the same genes. First DNA injection at 0 weeks, second DNA injection at 10 weeks, and recombinant vaccinia virus booster injection at 20 plus weeks, around 25 weeks from start of inoculation procedure. And now, an insoluble extracellular polymer of glucosamidase, a DNA vaccine, was designed to prevent dental caries, including the coding sequences for alanine and proline-rich peptide, as well as the C-terminal domain of Streptococcus glucosyltransferase. This C-terminal domain is necessary for the binding of the glucosyltransferase to the bacterial cell surface. This DNA vaccine therefore encoded two separate peptides, both of which facilitate the binding of Streptococcus cells to the tooth surface in an attempt to overcome the tendency of many DNA vaccines to induce only a weak immune response. The DNA vaccine construct contained two additional elements. First, the extracellular domain of cytotoxic T lymphocyte antigen 4, CTLA4, which binds to the B7 protein that is expressed on the surface of antigen-presenting cells, was induced in the construct. Second, the Fc region of immunoglobulin G molecule, which can bind to the Fc receptor on the antigen-presenting cell, was included in this construct. The use of both of these peptides was designed to specifically target the multi-domain fusion protein to immune system cells and thereby amplify the immune response and enhance the efficacy of the vaccine. A DNA vaccine designed to prevent dental caries included the coding sequences for alanine and proline-rich peptide, as well as a C-terminal domain of Streptococcus glucosyltransferase. This is just a repetition for dental caries because it is equally an important topic. And for those who might not have listened to it, this C-terminal domain is necessary for binding of the glucosyltransferase to the bacterial cell surface. This DNA vaccine therefore encoded two separate peptides, both of which facilitate the binding of Streptococcus cells to the tooth surface in an attempt to overcome the tendency of many DNA vaccines to induce only a weak immune response. The DNA vaccine construct contained two additional elements. First, the extracellular domain of cytotoxic T lymphocyte antigen 4, CTLA4, which binds to the B7 protein that is expressed on the surface of antigen-presenting cells, was included in the construct. Second, the region of an immunoglobulin G molecule. The use of both of these peptides was designed to specifically target the multi-domain fusion protein, its immune cells, system cells, and thereby amplify the immune response and enhance the efficacy of the vaccine. In fact, rabbits immunized with this vaccine, either intranasally or intramuscularly, displayed a significantly enhanced specific system and mucosal immune response compared to immunization with only the alanine and proline-rich peptide fused to the C-terminal domain of glucosyltransferase. Moreover, it was subsequently shown that this DNA vaccine could provide significant protection against dental caries in rats that were challenged with S. mutans and S. sabrinus, although the problem of delivering the DNA vaccine to humans still needs to be addressed so that its clinical efficacy can be tested. This is a very promising strategy that could be enormously beneficial to human populations. Figure 1219 is a schematic representation of a multi-domain protein encoded by a DNA vaccine Two domains, the CTLA4 extracellular domain and the immunoglobulin G IgG Fc region, are designed to target the other two portions of the molecule, that is, the glucosyl transferase, C-terminal domain, and the alanine-proline rich peptide to antigen-presenting cells. The diagram is self-explanatory. Take a screenshot and should help you. About attenuated vaccines, which is also explained. I'll just give you the notes. In some instances, genetic manipulation may be used to construct modified organisms, bacteria, viruses that are used as live recombinant vaccines. These vaccines are either non-pathogenic organisms that have been engineered to carry and express antigenic determinants from a target pathogenic agent, or engineered strains of pathogenic organisms in which the virulence genes have been modified or deleted. In these instances, as part of a bacterium or a virus, the important antigenic determinants are presented to the immune system with the confirmation that it's very similar to the form of the antigen in the disease-causing organisms. Although successful in some cases, purified antigen alone often lacks the native conformation and elicits a weak immunological response. Cholera. It is usually advantageous to develop a live vaccine because they are generally much more effective than killed or subunit vaccines. The major requirement for a live vaccine is that no virulent forms be present in the inoculation material. With this objective in mind, a live cholera vaccine has been developed or was developed. Cholera caused by the bacterium V. cholerae is a fast-acting intestinal disease characterized by fever, dehydration, abdominal pain, and diarrhea. It is transmitted by drinking water contaminated with the fecal matter. In developing countries, the threat of cholera is a real and significant health concern whenever water purification and sewage disposal systems are inadequate. Since V. cholerae colonizes the surface of the intestinal mucosa, it was reasoned that an effective cholera vaccine should be administered orally and directed to the structure. With this in mind, a strain of V. cholerae was created with the part of the coding sequence for the A1 peptide deleted. The strain cannot produce active enterotoxin. Therefore, it is non-pathogenic and is a good candidate for a live vaccine. Specifically, in this experiment, a tetracycline resistance gene was incorporated into the A1 peptide DNA sequence on the V. cholerae chromosome. This insertion inactivated the A1 peptide activity and also made the strain resistant to tetracycline. Although the A1 peptide sequence has been disrupted, the strain is not acceptable as a vaccine because the inserted tetracycline resistance gene can excite spontaneously, thereby restoring enterotoxin activity. Consequently, it was necessary to engineer a strain carrying a defective A1 peptide sequence that could not revert. A plasmid containing the cloned DNA segment for the A1 peptide was digested with the restriction enzymes CLA1 and XBA1, each of which cuts only within the A1 peptide coding sequence of the insert. To recircularize the plasmid, an XBA1 linker was added to the CLA1 site and then cut with XBA1. T4DNA ligase was used to join the plasmid at the XBA sites, thereby deleting a 550 base pair segment from the middle of the A1 peptide coding region. This region removed 183 of the 994 amino acids of the A1 peptide. Then, by conjugation, plasmid containing the deleted A1 peptide coding sequence was transferred into the V. cholerae strain carrying the tetracycline resistance gene with its A1 peptide DNA sequence. 5. Recombination, a double crossover between the remaining A1 coding sequence on the plasmid and the tetracycline resistance gene disrupted A1 peptide gene on the chromosome replaced the chromosomal A1 peptide coding sequence with a homologous segment on the plasmid carrying the lesion. 6. After growth for a number of generations, the extrachromosomal plasmids, which is unstable in V. cholerae, was spontaneously lost. Cells with an integrated defective A1 peptide were selected on the basis of their tetracycline sensitivity. The desired cells no longer had the tetracycline resistance gene, but carried the A1 peptide sequence with a deletion. A stable strain with an A1 peptide sequence containing a deletion was selected in this way. This strain did not produce active enterotoxin, but nevertheless retained all the other biochemical features of the pathogenic form of V. cholerae, that is V. cholerae, with an A1 peptide containing a deletion is a good vaccine candidate because the bacterium that synthesizes only the A2 and B peptides is as immunogenic as the native bacterium. When the strain was evaluated in clinical trials to test its effectiveness as a cholera vaccine, the results were equivocal. While the vaccine conferred nearly 90% protection against diarrheal disease in volunteers, it induced side effects in some of those who were tested. This strain may require modification at another chromosomal locus before it can be used as a vaccine. Salmonella species. Other attempts to engineer non-pathogenic strains of pathogenic bacteria that could be used as a live vaccine have involved deletions in chromosomal regions the code for independent and essential functions. At least 2 deletions are preferred because the probability that both sets of functions can be simultaneously reacquired is very small. It is assumed that a doubly deleted strain would have a limited ability to proliferate when it is used as a vaccine, thereby curtailing its pathogenicity while allowing it to stimulate an immunological response. Now, strains of the genus Salmonella cause enteric fever, infant death, typhoid fever, and food poisoning. Therefore, an effective vaccine against these organisms is needed. Deletions in a number of different genes have been used to attenuate various Salmonella strains. These mutations can be grouped into 3 basic categories: mutations in 1. Biosynthetic genes, 2. Regulatory genes, and 3. Genes involved in virulence. In addition, strains with more than 1 deletion have been constructed. For example, 1 double deletion strain has deletions in the ARO genes, which encode enzymes involved in the biosynthesis of aromatic compounds, and in the PER genes, which encode enzymes involved in purine metabolism. These double deletion strains, which can be grown on a complete and enriched medium that supplies the missing nutrients, generally establish only low-level infections, since their host cells contain only a very low level of the metabolites that they require for growth. Typically, their virulence is reduced by 100-fold or more. These attenuated Salmonella strains are effective oral vaccines for mice, sheep, cattle, chickens, and humans. Deletion of the DAM gene, which encodes DNA methylase, may be a highly effective approach to produce a virulent Salmonella strains. The DAM gene is a master switch that regulates the expression of 20 to 40 different Salmonella regulatory proteins. Thus, when mice were immunized with DAM-negative strains of Salmonella, they tolerated up to 10,000 times the normal lethal dose. Generally, pathogenic bacteria turn on many of the genes as briefly as possible to avoid detection and attack by the host's immune system. However, the DAM-negative strains, these genes are expressed for much longer periods, making it easier for the host immune system to detect and destroy the invading bacteria. I repeat, the DAM-negative strains. Because many other we come to this table later. Gut colonizing bacteria have DAM genes. If this approach with Salmonella turns out to be as effective as is expected, it may be possible to utilize a similar protocol with a range of pathogenic bacteria. Leishmania species. Although the human immune system can respond to infections by protozoan parasites of the genus Leishmania, it has been difficult to develop an effective vaccine against these organisms. Attenuated strains of leishmania are sometimes effective as vaccines. However, they often revert to virulence. Also, the attenuated parasite can persist for long periods in an infected but apparently asymptomatic individual. Such individuals can act as reservoirs for the parasite, which can be transferred to other people by an intermediate host. To overcome these problems, an attenuated strain of leishmania that is unstable to revert to virulence was created by targeted deletion of an essential metabolic gene, such as the one encoding dihydrofolate reductase thymidylate synthetase or synthase. One of these attenuated strains, Leishmania major E10-5A3, the 2-hydrofolate reductase thymidylate synthase genes that are present in wild-type strains were replaced with the genes encoding resistance to the antibiotics G-418 and hygromycin. For growth in culture, it is necessary to add thymidine to the medium that is used to propagate the attenuated but not the wild-type strain. In addition, unlike the wild-type, the attenuated strain is unable to replicate in macrophages in tissue culture unless thymidine is added to the growth medium. Importantly, the attenuated strain survives for only a few days when inoculated into mice. In that time, it does not cause any disease. Moreover, this period is sufficient to induce substantial immunity against leishmania in BALB/C mice after administration of the wild-type parasite. Since the attenuated parasite did not establish a persistent infection or cause disease, even in the most susceptible strains of mice tested, it is considered to be a strong candidate vaccine. Following additional experiments with animals, it should be possible to test whether this attenuated parasite is effective as a vaccine in humans. Well, this is at the 4th edition, so there could be a lot of changes by now. Please do your concerned research and follow up on each of my notes. If there are any additions, once I finish the entire subject, we'll get back to the additions and latest versions, right? Herpes simplex virus. As with other pathogenic organisms that have been developed as live vaccines, portions of the HSV gene genome have been deleted. Initially, it was thought that a strong immune response could be obtained only if the virus was able to replicate. However, several vaccines based on non-replicating viruses induce an immune response. Developing an avirulent HSV is important because subunit vaccines so far have been unsuccessful in inducing immunity against the virus. To prepare a safe and efficacious live HSV vaccine, 2 deletions in different locations in the viral genome were generated independently and then combined to form a double deletion virus. This strain is unable to proliferate in host cells, and the probability that both sets of functions can be simultaneously reacquired is very small. This replication defective strain induces protective immunity that can reduce acute viral shedding and latent infection. Vector vaccines. Now, vaccines directed against viruses. Vaccine of virus in the form of a live vaccine has led to the eradication of smallpox globally. Why? Vaccinevirus is a member of the poxvirus family. This completely sequenced virus has a double-stranded DNA genome that contains 187 kilobase pairs kb and encodes approximately 200 different proteins. Vaccinevirus DNA replicates within the cytoplasm of infected cells. Cytoplasmic rather than nuclear replication and transcription are possible because vaccinevirus DNA contains genes for DNA polymerase, RNA polymerase, and the enzymes to cap, methylate, and polyadenylate messenger RNA, mRNA. Thus, if a foreign gene is inserted into the vaccinia virus genome under the control of a vaccinevirus promoter, it will be expressed independently of host regulatory and enzymatic functions. The virus can infect humans and many other vertebrates as well as invertebrates. In addition to having a broad host range, vaccinevirus is well characterized at the molecular level is stable for years after lyophilization, freeze-drying, and is usually a benign virus. For these reasons, it is a strong candidate as a vector vaccine. The function of a vector vaccine is to deliver and express cloned genes encoding antigens that elicit neutralizing antibodies against pathogenic agents. Unfortunately, the vaccinevirus genome is very large and lacks unique restriction sites. Therefore, it is not possible to insert additional DNA directly into the viral genome. Of necessity, the genes for specific antigens must be introduced into the viral genome by in vivo homologous recombination. The DNA sequence coding for a specific antigen, such as HPCAG, is inserted into a plasmid vector immediately downstream of a cloned vaccinia virus promoter and in the middle of a non-essential vaccinevirus gene, such as a gene for the enzyme thymidine kinase. This plasmid is used to transfect thymidine kinase negative animal cells in culture, usually, chicken embryo fibroblasts that have previously been infected with wild-type vaccinevirus, which produces the functional thymidine kinase. Recombination between DNA sequences that flank the promoter and the neutralizing antigen gene on the plasmid and the homologous sequences in the viral genome results in the incorporation of the cloned gene into the viral DNA. Although the recombinant combination even is rare, the absence of thymidine kinase. Please take screenshots of these diagrams. Kinase activity in the host cells and the disruption of thymidine kinase gene in the recombined virus render in the host cells, resistant to the otherwise toxic effects of bromodeoxyuridine. This selection scheme enriches for cell lines that carry a recombinant vaccinevirus. The definitive selection of cells with a recombinant vaccinevirus is made by DNA hybridization with a probe for the antigen gene. Since thymidine kinase negative mutants of vaccinevirus arise spontaneously at a relatively high frequency of about 1 virus particle in 10 raised to 3 to 10 raised to 4, a selectable marker is often co-transferred with the target gene. This makes it much easier to distinguish a spontaneous thymidine kinase mutant from a mutant deliberately generated by homologous recombination. In other words, a virus with a spontaneous mutation would not carry the selectable marker, whereas a virus that underwent a homologous recombination would. The neogene, which encodes the enzyme neomycin phosphotransferase 2 and confers resistance to the kanamycin analog G418, is often used as a selectable marker. This gene, unlike some other selectable markers, is quite stable once it is inserted to the vaccinevirus genome. To avoid disrupting any vaccinevirus genes or the necessity of screening for selectable markers, a novel system has been devised in which every recombinant virus that can form a plaque will contain and express the target gene. The wild-type vaccinevirus contains a gene, VP37, that is responsible for the formation of plaques when the virus is grown on animal cell monolayer. Deleting the VP37 gene and replacing it with an E. coli marker gene creates a vaccinevirus mutant that does not form plaques after 2 to 3 days of growth in cell culture. Target genes are introduced into the mutant vaccinevirus by homologous recombination with a transfer vector that carries the VP37 gene as well as the target gene. If homologous recombination between the non-plaque forming mutant and the transfer vector occurs, the virus that can form plaques have acquired VP37 gene. Also, the target gene is inserted into the vaccinevirus genome and the selectable marker gene is lost. Since the VP37 gene has been deleted in the mutant vaccinevirus, it is impossible for this mutation to revert to the wild type. Therefore, every virus that forms a plaque carries the desired construct. This procedure is simple and straightforward, is applicable to the cloning and expression of any target gene, does not require any extra marker genes, and does not disrupt any vaccinevirus genes. A number of antigen genes that have been successfully inserted into the vaccinevirus genome and subsequently expressed in animal cells and culture These antigens include rabies, virus G protein, hepatitis B surface antigen, SINT-B's, virus surface proteins, influenza virus NP and HA proteins, vesicles stomatitis virus N and G proteins, and HSV glycoproteins. Several recombinant vaccinia-virus vehicles have been shown to be effective vaccines. For example, a recombinant vaccinia virus that expresses the HSV1-GD glycoprotein D gene prevents herpes infections in mice. Another recombinant vaccinia virus that expresses the rabies virus surface antigen gene was able to elicit neutralizing antibodies in foxes, which are major carriers of rabies in Europe. Has been used in the field for some time, including an area of approximately 10,000 square kilometers in Belgium. The vaccinia rabies virus glycoprotein recombinant virus vaccine that is presently on the market, RiboviriaKingdom, is a live viral vaccine containing 10^8 plaque-forming units, PFU, or live viral particles per dose. It is constructed by insertion of the DNA copy coding for glycoprotein G of a rabies virus strain which stimulates the thymidine kinase gene of a strain of vaccinia virus. Once the vaccine is ingested by a fox, the vaccinia virus begins to replicate and express rabies glycoprotein G, which stimulates the development of immune responses to the rabies glycoprotein. This results in the production of neutralizing antibodies against rabies virus in the immunized foxes. This immunity typically lasts about 12 months in cubs and 18 months in adult animals. The use of vector vaccines constructed from vaccinia virus also offers the possibility of vaccinating individuals against several different diseases, with one treatment. This may be achieved by using a recombinant vaccinia virus carrying a clone genes encoding a number of different antigens. The timing of the production of a foreign protein whose gene is carried in a vaccinia virus depends on whether a vaccinia virus promoter functions. During the early or late phase of the infection cycle. And the strength of the promoter determines the amount of an antigen that is produced. For the most part, late promoters for an 11 kilodalton protein P11 and the cowpox virus A-type inclusion protein PCAE have been used to achieve high levels of foreign gene expression. When genes encoding several different foreign proteins are inserted into one vaccinia virus, each is placed under the control of a different vaccinia virus promoter to avoid the possibility of homologous recombination between different portions of the virus genome that might have caused the clone genes to be lost. A live recombinant viral vaccine has several advantages over killed virus or subunit vaccines. First, the virus can express the authentic antigens in a manner that closely resembles a natural infection. Second, the virus can replicate within the host, thereby amplifying the amount of antigen that activates the release of antibodies from B cells, the immune response, and stimulates the production of T cells, cell-mediated immune response. A disadvantage of using a live recombinant viral vaccine is that vaccination of an immunosuppressed host, such as an individual with AIDS, can lead to a serious viral infection. One way to avoid this problem may be to insert the gene encoding human IL-2 into the viral vector. IL-2 enhances the response of the T cells of the immune system, enabling the recipient to limit the proliferation of the viral vector, and thereby decreases the possibility of an unwanted infection. If the proliferation of vaccine or vaccinia virus has deleterious effects in certain patients, it would be helpful to kill or inhibit it after vaccination. One approach is to create an interferon-sensitive vaccinia virus. Wild-type vaccinia virus is relatively resistant to interferon. Such a virus vector would be susceptible to drug intervention if complications from vaccination with vaccinevirus vectors arose. The basis of the resistance of vaccinevirus to interferon was not known until a vaccinevirus open reading frame K3L was found to encode a 10.5 kilodalton protein that has an amino acid sequence that is very similar to that of a protein of a 36.1 kilodalton host cell eukaryotic initiation factor 2A, EIF2A. The N-terminal regions of both of these proteins contain 87 amino acids that are nearly identical. Moreover, this shared sequence contains a serine residue, amino acid 51, which in EIF2A is normally phosphorylated by interferon-activated P1 kinase. When this serine residue on EIF2A is phosphorylated in interferon-treated cells, protein synthesis and therefore viral replication is inhibited. Thus, vaccinevirus may avoid inhibition by interferon because the K3L protein acts as a competitive inhibitor of E1F2A phosphorylation. Therefore, deletion of all or a portion of the K3L gene from vaccinevirus should make the virus sensitive to interferon. A K3L negative mutant of vaccinevirus was constructed by PCR mutagenesis of the K3L gene carried on a plasmid, followed by homologous recombination to replace the wild-type K3L sequences with the modified version. When the wild-type and mutant version of vaccinevirus were tested for sensitivity to interferon, the mutant was 10 to 15 times more sensitive to interferon than was the wild-type version. Reinsertion of the wild-type K3L sequence into the mutant virus restored the level of interferon sensitivity found in the wild-type. This indicates that K3L gene is indeed involved in the interferon resistance phenotype. A vaccinevirus. This work is an important step in the development of safer vaccinevirus vectors. Moreover, other interferon-resistant viruses may contain sequences comparable to K3L and therefore may be amenable to the construction of interferon-sensitive deletion mutants. Other comparable approaches to the creation of attenuated strains of vaccinevirus have been developed. For example, a strain of vaccinevirus constructed to have a mutation in the B8R gene, which encodes an interferon viral receptor, it is less pathogenic for mice than the parental viral strain. Currently, several veterinary vaccinevirus-based vaccines have been licensed, and clinical studies to test their efficacy in preventing a number of human infectious diseases are underway. The technology is based on a part of the development of an attenuated version of the vaccinevirus strain that has previously been used in the eradication of smallpox. To avoid any risk of the vector itself becoming a source of disease, some genetic information was removed from the virus genome so that the viral vector was highly attenuated. This attenuated virus has been used to express a number of viral antigens, with the expectation that the recombinant virus would be an effective live vaccine. Protection has been achieved by cloning glycoproteins from porcine pseudorabies virus, hemagglutinin, glycoproteins from equine influenza virus or a spike protein from the SARS virus, and the polyprotein of the Japanese swine encephalitis virus, and then vaccinating humans to prevent transmission of these viruses. Based on the success of these attenuated vaccinevirus vaccines, it has been proposed that this virus be considered a general delivery system for a wide range of proteins. For mass vaccination campaigns in developing countries, it will be advantageous to be able to deliver a live vaccine in a simple, expeditious, and cost-effective manner. In addition, with mucosally transmitted pathogens such as HIV, traditional vaccination routes may not induce mucosal immune responses sufficient to provide protective immunity. One possible alternative to traditional vaccination is aerosol immunization, which is potentially safer, easier, and less expensive to administer. To this end, researchers tested the abilities of two attenuated attenuated vaccinia virus-based vectors to be delivered effectively by aerosol immunization. In fact, it was found that aerosol delivery was both safe and effective, yielding long-lasting systemic and mucosal immune responses. When delivered to rhesus monkeys, this approach still needs to be tested with humans. However, it could offer an effective means of inoculating large numbers of individuals in the future. Although much of the work on the development of live viral vaccines has been done with vaccinia other viruses such as adenovirus, poliovirus, and varicella zoster virus are also being tested as potential vaccine vectors. Live attenuated poliovirus can be delivered orally, and such a mucosal vaccine which is directed to receptors in the lungs or gastrointestinal tract might also be useful against a range of diseases, including cholera, typhoid fever, influenza, pneumonia, mononucleosis, and rabies. However, the safety and efficacy of any apparently benign virus as a gene delivery and expression system must be firmly established before clinical trials are undertaken. Vaccine directed against bacteria. Since the discovery and subsequent widespread dissemination of antibiotics, only a modest amount of research has been done directed toward the development of vaccines for bacterial diseases. However, there are good reasons for developing bacterial vaccines. Not all bacterial diseases are readily treated with antibiotics. The use of antibiotics over the last 40 years has resulted in the proliferation of bacterial strains that are resistant to several antibiotics. Reliable refrigeration facilities for the storage of antibiotics are not commonly available in many tropical countries. It is often difficult to ensure that individuals receiving antibiotic therapy undergo a full course of treatment. Given the need to produce vaccines that will be effective against bacterial diseases, the question is which strategies are likely to be most effective. In instances where the disease-causing bacterium does not grow well in culture, the development of an attenuated strain is not feasible. For these bacteria, all alternative species must be used. For example, Rickettsia rickettsii, a gram-negative obligately intracellular bacterium that causes Rocky Mountain spotted fever, does not grow in culture. In this case, a cloned 155 kDa kilodalton protein that is a major surface antigen of R. rickettsii was used as a subunit vaccine and was found to protect immunized mice against infection by this disease-causing bacterium. Tuberculosis. Tuberculosis, one of the most important infectious diseases worldwide, is caused by the bacterium M. tuberculosis. The bacterium can form lesions in any tissue or organ, which leads to cell death. The lungs are most commonly affected. Patients suffer fever and loss of body weight, and without treatments, tuberculosis is often fatal. It is estimated that approximately 2 billion people are currently infected with the organism and that approximately 2 million to 3 million deaths a year result from these infections. Over the past 50 years, antibiotics have been used to treat patients with M. tuberculosis. However, numerous multi-drug resistant strains of M. tuberculosis are now prevalent in the United States among HIV patients infected with an antibiotic resistant strain of M. tuberculosis. There is a 50% mortality rate within 60 days. Consequently, a bacterial disease that was thought to be under control has become a serious public health problem in many parts of the world. Currently, in some countries, Bacillus calmette-guerin, BCG, an attenuated strain of Mycobacterium bovis that was developed between 1906 and 1919, is used as a vaccine against tuberculosis. However, the use of this vaccine has some drawbacks. First, live BCG cells can cause tuberculosis in immunocompromised individuals such as AIDS patients. Second, individuals treated with BCG respond positively to a common tuberculosis diagnostic test, which makes it impossible to distinguish between individuals infected with memb tuberculosis and those inoculated with BCG cells. For these reasons, the BCG strain is not approved for use in a number of countries, including the United States. In an attempt to determine whether a safer and more effective vaccine against tuberculosis might be developed, the extent of the immunoprotection elicited by purified M. tuberculosis bacterium is in liquid culture 6 of the most abundant of the approximately 100 secreted proteins were purified. Each of these proteins was used separately and then in combination to immunize guinea pigs. The immunized animals were then challenged with an aerosol containing approximately 200 cells of live M. tuberculosis, a large dose for these animals. The animals were observed for 9 to 10 weeks before their lungs and spleens were examined for the presence of disease-causing organisms. In these experiments, some of the purified protein combinations provided a slightly lower level of protection against weight loss, death, and infection of lungs and spleen than did the live BCG vaccine. Prominent among the proteins that provided protection was the M. tuberculosis major secretory protein, a 30 kilodalton transferase, also known as alpha antigen or antigen 85B. However, DNA vaccine encoding this protein was even less effective than the purified secreted protein. While this and possibly other M. tuberculosis secreted proteins might even actually be a part of a safe and efficacious vaccine for the prevention of tuberculosis in humans, it is necessary to develop a suitable delivery system for them. In theory, the optimal delivery system for an antigen that provides protection against tuberculosis should be 1. Able to multiply in the mammalian host, 2. Non-pathogenic, and 3. Able to express and secrete the protective antigen. Therefore, an E. coli mycobacterium shuttle vector that contained the gene for the 30 kilodalton protein alpha antigen under the control of its own promoter was introduced in 2 different BCG strains. Transformed cells produced 2 to 5.4-fold more 30 kilodalton protein than did non-transformed cells. In addition, despite the fact that the introduced genes were plasmid encoded and therefore potentially unstable, transformed cells continue to express a high level of 30 kilodalton protein after the vaccination of a test animal, in agreement with the hypothesis that the extracellular proteins of intracellular organisms are key immunoprotective molecules. Guinea pigs immunized with transformed BCG strains had significantly fewer bacilli in their lungs and spleens. In addition, there were smaller and fewer lesions in their lungs, spleens, and livers, and the survival of the animals was significantly increased compared with animals vaccinated with a non-transformed BCG strain. This is the first report of a vaccine against tuberculosis that is more potent than the currently available commercial vaccine. You would have much later versions right now. Things have transformed and developed much better now. Please update. This vaccine is currently in clinical trials. I'm just giving these notes for you as the basics. Use them as basics, build upon that, and make your own research for the latest updates in the concerned topic, and use them to the best of your exam and your benefit of a better career, right? And more of it's possible to prepare dried preparations of BCG in which individual bacteria form rod-like structures 1 to 4 micrometers long and 0.2 to 0.4 micrometers in diameter that may serve as the basis for live bacterial vaccine that is delivered as an aerosol, thereby facilitating the inoculation of newborn infants. Bacteria as an antigen delivery systems. Antigens that are located on the outer surface of a bacterial cell are more likely to be immunogenic than are those in the cytoplasm. Thus, one strategy is to replace a neutralizing antigen from a pathogenic bacterium on the surface of a live non-pathogenic bacterium. Flagella are made up of filaments of a single protein called flagellin. Under a microscope, they appear as conspicuous thread-like structures in the outer surfaces of some bacteria. If the flagella of a non-pathogenic organism could be made to carry a specific epitope from a pathogenic bacterium, protective immunogenicity might be easily achieved. This strategy was used to engineer a cholera vaccine. A synthetic oligonucleotide specifying an epitope of the cholera toxin B subunit was inserted into a portion of the Salmonella flagellin gene that varies considerably from one strain to another hypovariable segment. The construct was then introduced into a flagellin negative strain of Salmonella. This lecture will be over in a minute from now, and the continuation will be in the next lecture. Please make notes and use them to the best of your examination and to the benefits of your career. Kindly do not commercialize any aspect of these lectures or these screenshots that you take. Thank you so much. And I'll just sum it up now with a few words on the concerned subject. The epitope, which consists of amino acid residues 50 to 64 of the cholera toxins B subunit, was shown previously to elicit antibodies directed against intact cholera toxin. The chimeric flagellin functioned normally. Furthermore, the epitope was present at the flagellum surface. Immunization of mice was effective, and attenuated Salmonella strains can be administered orally, which would enable them to deliver a range of bacterial, viral, and parasite antigens to the mucosal immune system.